going to hit record. Cool. Is it recording? Was it recording? Now it's recording. Okay. All right, I'm showing you this first. Always do your attendance, always do your attendance, always do your attendance. Oh my God, you're killing me. Y'all are killing me. Attendance is almost over for the 6 weeks. I hope your grades are where you want them to be. Go away. We are still in the skeletal muscle tissue. I do plan on finishing it today. Do you need to go? Okay. And then I will lecture whole muscle contraction remotely, and y'all can just watch it at your leisure. And also the same for smooth muscle tissue, but we'll do muscles of the body together as a class as well. But also you can do it on your own to start learning the muscles of the body. All right, so there's that. That's what we're doing, muscles forever. Now, where's our PowerPoint? We stopped at slide 44. What we've been doing is going from the inside of the muscle cell to the very minutest part, outside to the myofibril. And today we're going to talk about the nerve impulse that comes from obviously somewhere else, the spinal cord or the brain, and how that excites the myofibril, causing the sarcomere, each individual sarcomere, to contract. That's the end game here. The videos that we watch in this section of the PowerPoint pretty much summarize the entire PowerPoint. So if it's just too much for you, make sure that you're watching the videos so they are, let me remember to share my computer sound so that they are, so they have that information at least. We have 4 steps to muscle contraction. You have a nerve that has to stimulate the muscle. The muscle has to be stimulated. You have an action potential. Going to show up there anyway. Then I want it to, I just want the big screen on my screen. That's what I want. What the hell is wrong with you? I hate you so much. All right, you know what? Ah, you don't have a choice now. And yet you're still not doing it. There we go. However, I need to exit this and turn off that noise. Let's go away. Thank you. Okay, finally. Sorry, nerve stimulation. Nerve has to come from somewhere. When that nerve gets to the cell that it needs to get to, it's going to cause something called an action potential. An electric current that has to generate in the cytoplasm of the cell it wants to excite. The action potential must be strong enough then to propagate along the sarcolemma or the cell membrane. I said cytoplasm, I meant cell membrane, apologies, at sarcolemma. And then the intracellular calcium levels must rise briefly. Steps 1 and 2 occur at a neuromuscular junction, a nerve-muscle junction. Steps 3 and 4 link excitation to contraction. So the nerve, the action potential is excitation, and then contraction. That is all happening because of calcium ions. Here are the steps. So it's actually very sequential. It's actually very step by step that things have to happen. An action potential has to arrive down an axon at a neuromuscular junction. So axons come from what? So you can say it louder. The neurons. Axons come from axons come from neurons. What other branching cell bits do neurons have?
Dendrites.
Very good, Cassandra. Yes, dendrites. So dendrites are the shorter branches that do cell-to-cell communication, and then you have axons that can go from the brain all the way down to the bottom of your foot is the longest one we have. That's not true. It actually comes from the lower spinal cord. Apologies. But that is still the longest one we have, lower spinal cord to the bottom of the foot, or they can be very short. In this case, they're probably coming from the spinal cord to another muscle. When that action potential arrives at the neuromuscular junction, acetylcholine, and this is the abbreviation for acetylcholine, which is a neurotransmitter, is released and binds to the receptors on the sarcolemma, which, if you recall, the sarcolemma is the plasma membrane of a muscle cell. We just give it a fancy name. Ion permeability of the sarcolemma changes. What that really means is we have a massive flip in ions where sodium floods in and potassium floods out. That causes depolarization to occur along the membrane. And the depolarization ignites another action. Well, it's actually the same action potential, but then it ignites it in the sarcolemma. We're going to stop there for a moment. So skeletal muscles are, of course, stimulated by somatic or body motor neurons, and they end at a neuromuscular junction or something called a motor end plate. It's the same thing, just different names. So here we have lots of little tiny writing, but follow along, please. Here is a myelinated axon. Do y'all know what myelinated means? Not so much something attached to it. You have demyelinated and myelinated nerves. What does it mean to be myelinated? It's one of the reasons you can't have a 0% body fat.
Is it like it's like sheath-like, myelin sheath?
They are called myelin sheaths. They're also called Schwann cells, Schwann being the name of the man who discovered them. And so what they are they are wrapped sheets of fatty tissue around the axon. And what it actually does is cause the nerve impulse to leap from bare bits of axon down the entire axon length. This allows nerve impulses to propagate faster than if they have to travel down the entire length of the axon. So instead they leap over these myelinated sheaths. Okay, so there we have myelination. So this is telling us that this nerve is causing rapid fire impulses, which we know because we know how fast our muscles move when we think about it. An action potential comes down, ends at this motor end plate, or also known as a neuromuscular junction. So we're going to blow up this little picture here. When the action potential hits the end of the axon, what happens is calcium, which we do need in the muscle, but first we need in the neuron, calcium floods into this vesicle area. This causes acetylcholine to be exocytosed into the synaptic space here between the nerve and the muscle cell. So that was very quick what I just said, but action potential ends at the end plate, the motor end plate here. Calcium floods into the terminal end of the axon, releasing acetylcholine into the interaxon space or the synaptic space here. The calcium, whoa, no, I guess we're going to stop there for a second. We're stopping there for a second. So synaptic space, synaptic cleft. The acetylcholine is contained in vesicles. It's a neurotransmitter. We know that. Y'all should know that already. And the sarcolemma of the muscle cells contains millions of acetylcholine receptors. So at neuromuscular junctions, which is what NMJ is standing for, it consists of an axon terminal, synaptic cleft, and junctional folds that have these receptors. So basically, acetylcholine is the chemical component of the axon potential. The electrical impulse has ended and switched to a chemical impulse, which will then actually be transferred back to an electrical impulse in the muscle plasma membrane or the sarcolemma. So this chemical impulse floods across these junctional folds and binds these receptors. It causes a conformational shape change on the receptors. And two ions then flood along their concentration gradients. So sodium floods into the cell and potassium floods out of the cell. And when this neuromuscular junction is at rest, then the cell is constantly pumping these ions against their concentration gradient so they can have this flooding event occur. This causes depolarization of the plasma membrane of the muscle cell. Okay. And all of this is ended, and I'll tell you more about that in a second. All of this is ended when acetylcholine esterase, an enzyme, comes along and breaks down the acetylcholine that is holding on to these receptors. And all this happens in milliseconds. I mean, in the time it has taken me to explain the process, it has happened billions of times in our bodies, just from normal fidgeting. Okay, so this is just repeating everything I just said. The nerve impulse arrives at the axon terminal, causing acetylcholine to be released into synaptic cleft. That is, after calcium floods that axon motor end plate. Acetylcholine diffuses across the cleft and binds with receptors on the sarcolemma. It causes a conformational shape change on those receptors that leads to electrical events. Ions are pieces of electricity. So sodium is electric, potassium is electric, that ultimately generate an action potential through the muscle fiber, and acetylcholine is quickly broken down by acetylcholine esterase. If it wasn't, you would become rigid and you would stop. Everything would stop. You'd stop breathing too, also as well, because your rib muscles would have contracted and they wouldn't be able to relax if acetylcholine esterase didn't do its job, and then you would die because you're not breathing. So here is our first video, repeating everything I just said again, but it's a different format. And I think once you watch the videos, it all just becomes very, very clear. Everything we've talked about up to this point. So watching a video.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction.
Or motor end plate.
This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
Cation channels.
Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
I mean, she's dry as a bone, but she knows what she's talking about. Also, she's computer generated cation channels. But everything there is exactly right. Also, I would like you to note, where is it? Watch that action potential that is generated along the sarcolemma after depolarization occurs travels down the T tubules. Do you remember what the T tubules were? It's part of a triad of the smooth endoplasmic reticulum cistern, and then a T tubule, and then another cistern. And what's held in those cisterns? Do y'all remember? I'm sorry. It's an ion we've already talked about that was important in the nerve. Now it's going to be important in the muscle cell. I'll give you one of three options because we talked about three of them: calcium, potassium, sodium. Lennon says sodium. What do y'all say online? Oops. Anybody? Bueller? Anybody? Am I talking to myself again? I do that all the time. Calcium. Calcium is going to be the thing. I didn't know I clicked the button. Sorry, I was trying to look at people. So that nerve impulse, the whole point of it, blah, 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 blah. This is just repeating everything I've said twice, is to depolarize a sarcolemma. So the plasma membrane of the muscle cell. When it's at rest, it's polarized, which means that it has a negative and positive charge across the membrane like it's supposed to, and it's, you know, relatively equal. Inside of the cell is negative compared to outside because of all those sodium ions that we have pumped to the outside of the cell. When the end plate potential happens and we flood that cell with those sodium ions, it depolarizes. This is also, by the way, exactly what happens in a nerve cell as well. So this is a small picture into the next chapter in this book, which is, or not book, but this class, which is, help me, what's next? What's the nerve? What is that? The nervous system. Yes, thank you. And then repolarization, of course, will have to occur or your muscle will never relax, ever. So, blah, 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 blah. We've already talked about it. You know this now. This is old news. Acetylcholine is released, causes chemically gated ion channels to open, sodium diffuses in, potassium diffuses outward, but it's much more sodium than potassium. This causes that polarization to not be a thing, and so you depolarize the cell to a more positive electricity side, and that results in local depolarization called an end plate potential. So here you go. So so we're going to have a wave of depolarization. Now, I would like to say already that it can stop here if the signal isn't strong enough, like a twitch. Have you ever just twitched? That was when the polarization or depolarization signal wasn't strong enough to actually contract the muscle. It happens when you're falling asleep or when your mind is drifting type of thing. So it's actually a response to something that's happening in your brain. So we generate a wave of depolarization. It has to be strong enough. There has to be a threshold it has to hit to go along the entire sarcolemma and cause contraction of the muscle. So we've depolarized and created another, but also the same action potential. I shouldn't say another, because what we have done is taken the electric action potential of the nerve, turned it into acetylcholine chemical signal, and now we're turning it back into an electric signal. So it's the same wave, it's the same potential. If enough sodium floods in and we hit that threshold, then the action potential is unstoppable and will lead to muscle fiber contraction. And then once it spreads across the sarcolemma from one sodium channel to the next, it causes that whole, it's a wave of depolarization. It has to wave across the entire muscle fiber. We repolarize by closing the channels and pumping sodium out and bringing potassium back in. You have a refractory period. Now, for humans, it's milliseconds. It's not even milliseconds because that's a countable thing for us. But there is a very short time frame that is not real by our standards, but down at the muscle cell level, it's real, where the muscle fiber cannot be stimulated again until everything is repolarized all the way along the muscle fiber. That's called a refractory period. It happens faster than our brains can think about it. Does that make sense? Like we're in the milliseconds type of thing. So for the action potential to occur, sodium has to keep flooding in, but then down further it has to flood in, but then down further it has to flood in. So it's a wave of depolarization. And then we have to go back and reset. Sodium on the outside and potassium on the inside so that we could do it again. Our threshold amount is plus 30 millivolts. That is important for you to know, but more so just know what the graph looks like. We have a sharp peak, it goes away really fast, and then we are set to repeat. Okay, so look, you go from negative 90 in a polarized cell and you shoot it up to positive 30 over for three milliseconds. So we're at 3 here, 5. So over 5-ish milliseconds, and then you're back to normal. When you excite and depolarize the sarcolemma, the main thing that happens is that depolarization wave travels down the T tubules and it actually causes a conformational change from the sarcoplasmic reticulum, in the sarcoplasmic reticulum, that releases massive amounts of calcium into the cell. Follow along with the words. Calcium is going to go bind tropomyosin. Troponin is going to now move tropomyosin away from the actin heads, and myosin can now bind to actin and pull and bind and pull and bind and pull, so the sarcomere can contract. But then all the sarcomeres in that muscle cell or myofibril need to contract as well. The action potential is brief. Here, if you could star this. I have those abilities, don't I? The action potential is brief and ends before contraction is seen. So this isn't something that's happening constantly. Like the action potential is not happening constantly. It just waves through the cell and goes on. Okay. So the action potential propagates along the sarcolemma and down the T tubules. That's where we need it to be. Calcium ions are released from that sarcoplasmic reticulum cisternase on either side of the T tubule, because voltage sensitive tubule proteins change shape. That shape change allows the calcium to flood into the cytosol. So let's look at this video that is repeating everything I just said.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So make sure you always have enough calcium in your body. And this is the whole point of everything. This is why we have terminal cisternae full of calcium, is to do cross-bridge cycling. Now we are going to the sarcomere level, where those actin and myosin filaments are now going to interact with each other. When that cell has low calcium concentration in the cytosol, tropomyosin blocks the active sites on actin. We need tropomyosin to move so that myosin heads can attach to actin. If it doesn't happen, your muscle fiber remains relaxed. So, voltage-sensitive proteins in the T-tubules change shape, causing sarcoplasmic reticulum to release calcium into the cytosol. That calcium now binds to troponin. Okay, well, it binds to troponin. I've had it wrong the whole time. But when troponin is bound, it changes shape and moves tropomyosin away from those binding sites on the actin. Here, they're calling it myosin binding sites, but please remember, we're talking about the actin. The actin is being exposed. Now, myosin heads can bind to the actin. That is called a cross bridge. Cross bridge. Cross bridge. I don't know what I'm doing. Cross bridge. I'm really bad at this, but I'm gonna keep trying. The cycling is initiated, which means the myosin binds, releases, binds, pulls, releases, binds, pulls, releases, all while using ATP, by the way, causing sarcomere shortening and muscle contraction. When nerve stimulation ceases, calcium is pumped back into the sarcoplasmic reticulum and contraction ends. Oops. Clear, go away. So, we are at the sarcomere now. Remember, a myofibril is hundreds to thousands of sarcomeres. But calcium binds to troponin, which is the larger but lesser amount of protein that's blocking the actin. That causes a conformational shape change in the troponin, which moves the tropomyosin away from the binding sites. So now these myosin heads, which remember are the light chains of the myosin, can bind to the actin thin filaments. Using ATP, the myosin then moves slightly forward. Now you have thousands to millions of myosin heads doing this all at once. So that slight movement is actually rather powerful in terms of all the heads moving. And we have excited the cell and we are now contracting. So that coupling is over. Of course, there's going to be 4 steps to talk about in cross-bridge formation. You got a myosin binds to actin. Then you have a power stroke using ATP. Well, after ATP is used, you have a power stroke. Then a new ATP binds and the myosin head detaches to be able to do another power stroke. There's a video for this, but there's also this fun little circle diagram. So myosin attaches to actin using, well, ATP is ready to go. A power stroke occurs using that ATP. It's done. Now you got ADP and inorganic phosphorus. Then another ATP binds to cock the myosin head back into position to do another power stroke. Does that make sense? Okay. Here is the beautiful third and last video that just kind of summarizes everything I've talked about for 76 slides. Did I push play? Good.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Things you never knew. That's how much it takes to contract a muscle. Every single muscle in your body, every time you contract it, nerve impulse, acetylcholine, depolarization, calcium flooding, troponin, tropomyosin moving, myosin binding to actin, cross-bridge cycle over and over and over until there's no more calcium. And then the muscle relaxes. Crazy. This is our last slide. There's some, if things don't go right, you can, well, die for one. But rigor mortis is something people like to talk about a whole lot. Rigor mortis is actually a very short-lived event post-mortem or after death. But what happens is the myosin heads bind up to the actin, but then can't release. So because calcium cannot be pumped back into the sarcoplasmic reticulum because your cells are dead. So the results in a cross bridge formation that's never released, but the muscles will stay contracted until muscle proteins break down, which happens. So you go into rigor mortis about 3 to 4 hours after death, but about 12 hours after you enter rigor mortis, you actually begin to leave it. So you don't stay stiff forever. It's actually a really short-lived event in terms of postmortems. There. Now, I would like to point out, this is a very long PowerPoint, and I do not care that I have taken 3 class periods to go through it, but literally, if you understand the 3 videos, then you understand the entire PowerPoint. Okay? So if you need to review, then you're going to review the videos, unless you need to take better notes in your notebooks for whatever. Your lecture exam is going to be majority this PowerPoint. What? No. Sorry, trying to get back to your home screen. It's going to be a this PowerPoint that we just finished and naming the muscles. That's going to be a majority of your exam. We just have some extra information for you to know in terms of being a well-rounded individual in anatomy and physiology class. Okay, thank you so much to the 5 of you who have joined us, and remind Fatima and Lennon's right there, Thomas, to watch this video. Cool? Yeah, you powered through that. I saw you. You were good. All right, bye. Bye. Bye.
Bye.
Bye, Cassandra.
Bye, Miss Moore.
Bye, Pedro. Bye, Lennon.